Welcome to CTUX Research Training Videos. In this video, we're going to be going over how our Saxitoxin ELISA works. Think of this as background information on the technology and overall concepts of the method. The assay is an ELISA, which stands for Enzyme-Linked Immunosorbent Assay, and there are two key terms in this acronym. The first is enzyme. Enzymes are biological catalysts, and in this case, it's a protein that speeds up a reaction. It's the enzyme that causes the color change that allows you to see whether or not there is toxin in your sample. The second part is immunosorbent or immune. Think of your immune system. Immunosorbent refers to antibodies that attach to antigens, and for this assay, the antigen is the toxin. To break this down, let's start with antibodies. By this point, post-COVID pandemic, most people have heard of antibodies and know that they are part of your immune response to a pathogen. Antibodies are made by B cells, pictured here. B cells, upon activation, become antibody-producing factories, and they pump out tons of these little proteins as part of a coordinated response to a threat, typically an infection like a virus. Every threat or infection is made up of many different types of antigens, which are substances that cause the body to mount an immune response. Antibodies are one of the tools your body uses to fight off infections, and they are very, very specific to the type of antigen that raised the immune response. They can even be very specific to the small piece or epitope of an antigen that raised the immune response. This fact will become important later when we talk about our Saxitoxin antibodies, so keep that in the back of your mind. They are structurally similar to a Y shape with two antigen binding sites. And again, it's very specific to one antigen. In this picture, this antibody is only going to bind to that one yellow antigen because of that very specific shape. It's not going to bind to other antigens or even other parts or other epitopes of the same antigen. The second part is enzyme. Enzymes are almost always proteins that catalyze a reaction in a biological system. This is a schematic by OpenStack's college biology textbook, which shows how the process works. An enzyme works on a substrate, catalyzes the reaction, and releases the products. The enzyme used in most ELISA platforms is horseradish peroxidase, or HRP for short, which works on tetramethylbenzidine, or TMB for short. And when these two get together, they form a beautiful blue color. Now, these reactions are kinetic, and they will progress over time with a light blue color in the beginning developing to darker and darker blue. So we most often stop the reaction at a point where our samples fall within our standard curve. That part is really critical for valid ELISAs. And we stop it by adding acid. The acid denatures the enzyme protein and turns the color yellow. Here's an overview schematic of how our ELISA works. It's a competitive ELISA, which means that the toxin in a sample will compete for antigen binding sites on the Saxitoxin antibody with a colorimetric label, which for our ELISA is toxin conjugated to HRP. So let's go through the ELISA protocol. We're going to start with a 96-well plate that has been coated with what we call capture antibody. The capture antibody is stuck to the plate, and it recognizes the non-antigen binding end of our Saxitoxin antibody to capture it to the plate, hence why we call it a capture antibody. For our example here, we are going to designate one well as a high toxin concentration, like what we would use in our highest standard, or a sample that would be very toxic. We'll also designate a low toxin concentration, which would be our lowest standard, or a sample that is not very toxic. All right, let's clear those up. The first step is to add our Saxitoxin antibody. As you can see here, there is an abundance of antibody, where some are captured by the capture antibody. They settle down and get stuck to the plate, but there is still some floating around in the liquid as well. Next, we add our sample. This can be our standard curve, from low to high concentration of Saxitoxin. It can be our zero or vehicle control or no toxin, or it can be any of our unknown samples. On the right, you see that there are not very many. Only one is captured to the plate over here. However, on the left, there are many pieces of toxin in our well, since again, it's a toxic sample. So many of those bind to the saxitoxin antigen binding sites of our saxitoxin antibody, the green antibody. Next, we'll add our colorimetric label, or again, HRP conjugated saxitoxin. There is an equal amount in each well, but for the right well, most of them are going to settle down onto the antigen binding sites of the antibody because there are more open sites. On the left, more are taken up by the toxin in the sample, so fewer colorimetric labels are able to bind. They are outcompeted for binding sites by the toxin in the sample, and hence why we call it a competitive ELISA. Only some can bind to the sites, and whether that's toxin or colorimetric label depends on how much toxin is in your sample. Now, all of this is a bit hard to see because all the components are still in the solution. So the last step we do is to wash off everything that is unbound to the plate. Anything that's not stuck, that's not captured, will wash off. That way, the only thing that's going to react with the substrate is what has been captured by the assay. Next, we add that TMB substrate, which, if you'll remember, the enzyme catalyzes the reaction of the TMB substrate. If there's enzyme in the solution, as in the HRP conjugated saxitoxin on the right, then the solution will turn more blue. If there is little to no enzyme captured, because all of the antibody binding sites were outcompeted by toxin in the sample, as is on the left, then the solution turns less blue, if at all. Sometimes it's just clear. This reaction will keep going, and if you left it out for a long time, theoretically all of the wells would eventually turn the same color of blue, regardless of whether or not there's toxin in the sample. So we will stop the reaction by adding acid, and the acid again denatures the enzyme, or stops it, and it turns our solution yellow. It is very important to stop the reactions when the color of your sample is within the colors of your standard curve. You can watch our data analysis training video for more information and visualization as to why this is so important. But the short answer is that the results are only going to be valid for sample colors and sample concentrations that fall within the range of the colors of your standard curve. So why is saxitoxin detection for ELISAs so problematic? There are two major parent compounds to the paralytic shellfish poisoning toxin family, both of nearly equivalent toxicity and both which are prevalent in the environment and in seafood. Most commercially available immunoassays, so ELISAs, microplate assays, lateral flow assays, assays in a box, etc., are all based on an antibody that detects saxitoxin but not neosaxitoxin, or doesn't detect neosaxitoxin as well. The only difference between these two compounds is this one OH group. But that's enough to make a conformational change so that the saxitoxin antibodies of those kits do not recognize it, or again, don't recognize it well. Remember back when we talked about antibodies and how they are very specific to even pieces or epitopes of antigens. Because of this, those antibodies have very little cross-reactivity or ability to capture neosaxitoxin, even though it's a major component of the PSP toxins. The poor cross-reactivity with neosaxitoxin compounds the detection problem because it is the backbone for half of the PSP toxins, roughly. Here are just a few of the 20 to 50 structurally related compounds and toxins in the PSP family. Some are very toxic and some are not. But regardless, if an antibody only detects the saxitoxin side of the family, it will miss or have very little cross-reactivity, very little ability to detect the other half, which is a problem when trying to detect these toxic samples when neosaxitoxin and related compounds are prevalent in the environment. However, Cetox's antibody does have cross-reactivity, in fact, very good cross-reactivity with neosaxitoxin, which is the benefit over those alternatives. It can detect both saxitoxin and neosaxitoxin about the same. It can detect the GTXs with the saxitoxin backbone, 2 and 3, as well as the GTXs with the neosaxitoxin backbone, 1 and 4. And it has some ability to detect the decarbamylated saxitoxins. All of this was published in our 2019 paper, which is in the open access journal Toxins, and here's the DOI if you'd like to go and read more about it. Another thing that we worked on in that toxin paper was how to improve the detection efficiency of the GTXs. So naively, or without any modifications, cross-reactivity of our antibody with the GTXs is about 15%. But we found that we could improve it up to 90% with just a simple extra processing step using L-cysteine. The L-cysteine digestion is a step during sample processing that can be done to essentially make the GTXs look like Saxitoxin or Saxitoxin to the antibody, thus improving recognition and binding. Using L-cysteine, the STX ELISA resulted in very close alignment with the measurement of PSP toxins, including both parent backbones, to the results found from high-performance liquid chromatography. Now, this is important because of the relative toxicities of the various PSP toxin family members. This data in this table was compiled from the literature, and as you can see, the top 6 toxins, so Saxitoxin, Saxitoxin, and the GTX 1 through 4, can have nearly equivalent toxicities. If you're only measuring the Saxitoxin backbone, or in this case, GTX 2-3, then you're missing measurement of 3 key toxins that may be in the sample, which could result in a false negative result. In other words, your test could say that a seafood sample is safe when it is, in fact, not safe. To summarize, the STX ELISA is based on an antibody that detects not only Saxitoxin, but also Saxitoxin. It has cross-reactivity with the important or toxic family members of the STX backbone, and also of the Neo-STX backbone. Finally, with an easy addition of L-cysteine in the sample processing steps prior to running the ELISA, you can improve detection of the GTX family members for both parent backbones that might be in your sample, as we demonstrated in our 2019 paper. If you have any questions about this or how our ELISA works or how our antibodies work, please feel free to reach out to us. We would love to hear from you, and we hope that you found this video informative.